So the next important property of the plasma membrane exhibited as we discussed is the osmosis process. So let us now discuss the osmosis process in detail. Now what is this osmosis process? Osmosis can be defined as the movement of water molecules from its higher concentration area to the low concentration area through a semi-permeable membrane. Okay? So to understand it better, suppose if I make a tank and I keep a semi-permeable membrane in between. So in the middle I keep a semi-permeable membrane, and this side I keep a solution, and this side I keep plain water. Okay? So this is water and this is salt solution. Okay? So this side I have salt solution and this side I have kept the plain water. Now what the principle of osmosis says, if two different solutions are separated by a semi-permeable membrane. So this is a semi-permeable membrane. Okay, so this is a semi-permeable membrane which separates these two solutions. Then what is going to happen for the movement of water molecule or for the diffusion of water molecule? Now the diffusion of water molecule is termed as the osmosis process. Now what does the osmosis says? The water molecule will diffuse from its higher concentration area to its low concentration area. That its means what? The its means the water's higher concentration area to water's low concentration area. Suppose plain water is here and salt solution is there. Then whose water concentration is more? If solute is mixed, water concentration is definitely less. If it is a plain water, then water concentration is maximum. So now this particular part is the higher concentration area and this particular solution is the low concentration area. So what is going to happen? Slowly the water molecule will start moving from the plain water side to the salt solution side. Up to what level they will reach? They will diffuse till the concentration of both the sides are same. Clear? So till it becomes highly diluted, till that point the water will keep moving into the other side of the solution. Clear? So from its higher concentration area to its low concentration area. This is the process of osmosis. So depending on osmosis process, we can divide solutions into three major categories. Now what are the three major categories? The three major categories are solutions can be, if I write solution is of three major type. Okay, one is the hypotonic, second is the isotonic. Okay, second is the isotonic and the third one is the hypertonic. Okay, so hypertonic, isotonic and the hypotonic. Now which type of solution we will say it is hypotonic? Suppose if I just draw a vessel here. If I draw a vessel here, a vessel containing salt solution, and if I keep an animal cell inside, here inside the solution. Now suppose the concentration of water in the cytoplasm is less than the concentration of water in the outside medium, right? So hypotonic solution or hyper or isotonic is always a comparative term. We are comparing two different solutions. Suppose the concentration of water inside the cell is less than the concentration of water outside the cell. That means the container, the solution where the cell is put, the solution contains more water than the cytoplasm of the cell. Then what is going to happen? in what direction the water will move? The water will move definitely from its higher concentration area to low. So the water will start moving into the cell from the surrounding. Clear? This experiment you can try at home by using the dry grapes. Okay, so dry grapes you can use salt solution, you can use a similar type of concentration isotonic solution, you can use a very mild solution and a very thick salt solution, and you can observe the different type of changes that happens with the dry grapes. Clear? Okay, so this type of solution we call as hypotonic, when the concentration of the solution, the water concentration of the solution is higher than the concentration of water in the cytoplasm. So such type of solutions are called as hypotonic. Suppose if I draw one more again, this solution, the cell is here and the concentration of water is exactly the same as with the outside medium as well as the inside cytoplasm, right? So whatever water concentration is here in the cytoplasm, the same water concentration is now there in the surrounding medium. So such type of solutions will be called as the isotonic solutions. Why? Because there is no net movement of the molecules. Net movement means there is movement of molecules, but how much water is moving out of the cell to the solution, that much water is moving inside the cell from the solution. So entry and exit both are equal, so net movement is 0. Clear? So there is movement, but movement is equal inside the cell and outside the cell. So those type of solutions are called as the isotonic solution. Now the last one is the hypertonic solution. Suppose the solution which is kept in the container contains more solute, less water molecule. It is more concentrated solution, right? So I have mixed more salt or more sugar in that. It's a very thick solution. Less water, more solutes, right? So solvent is less, solute is more, and the cell which is kept here, that has more water content in comparison to the solution outside. So in that case, what is going to happen? Suppose the cell has more water content in comparison to the outside solution where it is kept, then where will the water move? What is the direction of water movement? Definitely the water will start moving out of the cell to the solution. Why? Because as per the rule of osmosis, the water will move from its higher concentration area to the low concentration area. Clear? So this type of solutions where the water concentration is less in compared to the cell cytoplasm, they are called as the hypertonic solutions. Clear? So 3 different types of solutions as per the principle of osmosis. So what are they? The hypotonic, isotonic and hypertonic. Clear? So in the next video, we will be discussing about the other parts of the cell.